So this morning we are going to be looking at culture media. Culture
Media.
When you talk about culture media, has any of you heard of culture media before? Can anyone help us define culture media? You are scratching your head. Okay, have you heard of culture media before? Hmm? Alright. When you talk about media, to culture, let's start from culture first. To culture is to what? To grow organisms. It's not the culture that you know about, like the way people live, the one you know in social study is different from what we are talking about. To culture in microbiology is to be able to grow microorganisms. It can be bacteria, it can be fungi, it can be viruses, it can be microalgae. You grow it in large quantity for either research purpose or for commercial use. Are you with me?
Yes.
So, you need specific mixture that contains nutrients. That's what the media is now. So, culture media is a mixture that contains specific nutrients. Specific nutrients. What do I mean by specific? Specific in the sense that there is a measurement and there are certain things that are used to prepare culture media. You use them to grow the kind of microorganisms that you want to work on, either to research on or to grow for commercial use. Now, we have what we refer to as agar. Agar. Agar is simply a solidifying agent. It's simply a solidifying agent. Now, it does not support the growth of any organism. Are you with me?
Yes.
You see that, for example, you prepare pap. You are trying to prepare pap, you mix the fresh one, and then you add hot water. And then you discover that, when you prepare it very thick, discover that you are trying to stir it. You may be unable to stir it because of the way you prepared it. Are you with me?
Yes.
That is how agar is. But agar will solidify it to the point that you can't even fall off a plate. So that if agar is in any media, you prepare it and it solidifies. When you turn the plate, it will not fall off. Are you with me? So that takes us to, so agar is simply a solidifying agent. It is what? A solidifying agent. It doesn't support the growth of any organism. It just helps you to solidify so that you can grow to have the morphology or colony of the organism that you want to grow. Are you following? So we have forms of culture media. Forms of culture media. We have three basic forms. The first one is the solid media. It can be in solid form. It can be in semi-solid form or media. Then thirdly, you have liquid media. Now, these are the forms of culture media that we have. The first one is the solid media. You prepare solid media to grow so that you can be able to see the colony of the organisms visibly. And as the name implies, it will solidify. That means you need to add the agar in large quantity. Are you following? So that it will solidify to the point that when you introduce or inoculate organisms into it, it will be able to grow the organism and show you the features, the morphology, the colony of the particular bacteria that you want to grow. Are you with me? The second one is the semi-solid media. That one, you add the agar in a minute quantity. You can use 0.2 can use 0.2, just add it small, ranging from 0.2 to 1 gram, so that when it is solidifying, it will not be too thick, it will not turn to solid. Are you with me?
Yes.
Now it will be like maybe you mix pap, not very thick, and then you added water. It will not be solid and it will not be liquid. Do you understand? Yeah, that's like semi-solid. And you use it to grow profuse growth. Use it for profuse growth. Then you have liquid media. Liquid media is also for profuse growth. That one is in liquid form. It's in liquid form. When you look at it, it's liquid. Are you with me? So these are the three forms of culture media. Now that takes us to types of culture media. Let's look at types of culture media. We have 6 basic types of culture media in microbiology. We have the basal media. Yeah. We have the basal media. We have the enriched media. We have the differential media. We have the selective media. We have transport media. And number 6, we have the storage media. Not somebody come and be writing this one for me in the exam. Let me better correct it. I hope you understand. Alright, so these are the 6 basic kinds of culture media that we have that is used in microbiology. Do you understand me? Yes. Now the first one is the basal media. Basal media can also be referred to as general purpose media. Can refer to as what? General purpose media. It is basal in the sense that it doesn't need enrichment. You don't enrich basal media. You can use it to grow different kinds of, for the ones for bacteria, you can use it to grow different kinds of bacteria. It is not specific and you don't enrich it. Are you with me? You don't add any other extra thing to it. It can be used for so many, to grow so many organisms. Then the second example of basal media, you have nutrient agar. Example, you have nutrient agar. This one is largely and greatly used in microbiology for the growth of both gram-positive and gram-negative bacteria. Are you with me? Yes. Then number 2, you have the enriched media. You have the enriched media. As the name implies, enriched, it means you added something as a source of more protein for the organism to be able to grow well. You enrich it. Are you with me? Yes. You can enrich media by adding blood. You can enrich media by adding air to ensure that there are basic nutrients available for this organism. So an example of enriched media, you have blood agar. You have blood agar media. Number 3, you have differential media. As the name implies, differential media differentiates. It normally has a specific preparation that contains a particular color. It will not differentiate the organism you are trying to grow amongst other organisms. Are you with me?
Yes.
For example, of this differential media, you have McConkey. McConkey, this is how it's spelled. McConkey agar. It differentiates gram-negative intestinal organisms. Are you with me?
Yes.
It is majorly used to grow gram-negative organisms. It's not like this basal media that can be used to grow both gram-positive and gram-negative. This one is basically used to grow gram-negative organisms, particularly those that are of the colon, those that belong to the family Neissereaceae. When you talk about this family of bacteria, you have bacteria like Proteus, you have bacteria like Klebsiella, you have bacteria like Salmonella. Are you with me?
Yes.
Salmonella typhi, and so on. Those are the organisms that are gram-negative that when you grow them, when you want to use this media to check them, you will see them growing, appearing pinkish in the background, depending on how the manufacturer prepared it. But the common one that we have, it will grow in a pink background. Once you see it, you know that it's a gram-negative organism. So for you to know the actual organism, whether Salmonella, whether Klebsiella, whether Proteus, whether Shigella, and so on, you now do biochemical test. Are you with me?
Yes.
Alright. Now the number 4 is selective media. As the name implies, it selects the organism that it wants to grow. And then the others will not grow. Makonki Egger does that work too. Another example is SSA Egger. SSA. This is Salmonella Shigella Egger. Salmonella Shigella Egger. Salmonella Shigella Egger. What this means is that you know Salmonella typhi are the causative agent of typhoid. And then you know Shigella dysentery that causes dysentery. These are part of the family I made mention of. They are under this Neissereaceae. Are you with me? They are all gram-negative. So by the time you use this SSA agar as a selective media to grow organisms, any organism that is not Salmonella and is not Shigella will not grow in that media. Are you following?
Yes.
But take for instance if it's a basal media, whether Salmonella or Staphylococcus or Streptococcus or Proteus, all of them will grow. Are you following? But if it's this one, for example, if it's this SSA agar, any organism that is not Salmonella and is not Shigella will not grow on that media. It is selective. That is what it means. Now you have number 5, you have a transport media. You have a transport media. As the name implies, it is used to transport organisms from the point of collection to the laboratory. Are you with me? Example, you have the stout media. Stout. And then you have peptone water too. You have peptone water. Transport media does not actually grow organisms, so are you with me? Use it to transport the organism from the point of collection, the sample from the point of collection to the laboratory, so that it will be active before you grow it. Are you with me? Then the last one is the storage media. As the name implies, they are media that are used to store organisms for a very long time. Remember I told you that some of these organisms, you grow them for commercial purpose. You grow them for what? Commercial purpose. So, if for example you did not sell them off at the time you grew them, these are the media that you use to store them before you can actually sell them. Alright, so these are the 6 basic types of cultural media that we have.